Thank you to everyone who's joined. Hi, and welcome. My name is Ellie Arnold, and I'm the training specialist at Stem Cell Network. As this session's moderator, it's my pleasure to welcome you all to this virtual session, Scientific and Investment Readiness for Aspiring Bioentrepreneurs. While we meet today on a virtual platform, I'd like to begin by acknowledging the Indigenous peoples of all the lands that we're on today. The work of Canada's Stem Cell Network occurs from coast to coast on the traditional, ancestral, and unceded territory of many Indigenous peoples and nations, including the Anishinaabe Algonquin Nation, where SCN is headquartered. SCN honors, recognizes, and respects these nations as the traditional stewards of the lands and waters where we work and meet. SCN is a not-for-profit organization supporting stem cell and regenerative medicine research through training, knowledge mobilization, and funding opportunities that advance the development of stem cell and regenerative medicine innovations. We're excited that you've joined us for today's session, which will aim to help you better understand the commercialization pathway for stem cell and regenerative medicine technologies and how you can set your research program up for commercial and investment success. This is just one of the many courses, workshops, and symposiums that we'll host throughout the year as part of our training strategic pillar. So I encourage everyone to go onto our website at stemcellnetwork.ca and check out those opportunities as they come up through the year. Before we get started, I just have a couple of housekeeping notes to go over. The session is being recorded, and the session will run for approximately an hour and a half until about 1:35 p.m. Eastern. Each speaker has prepared a talk about scientific and investment readiness. First, we'll have Chantel Martin give an overview of the commercialization pathway. There won't be any Q&A for that portion, as Chantel will be back to give a second talk later in the agenda. And then each of our three speakers will give a talk with about 5 minutes of Q&A. And you can put your questions in the Q&A box at the bottom of your screen, and I can pass them to the speakers. With that, I'd like to introduce and thank our speakers, Gregory Block, Matthew Jeffers, and Chantel Martin. Thank you all for being here today. And our first speaker will be Dr. Chantel Martin, who will provide an overview of the commercialization pathway from the research lab to an approved product. And again, this part will just be about 15 minutes. We'll do no Q&A, and then each speaker will have a 20-minute talk after that. Dr. Chantel Martin is a PhD-trained scientist from the University of Ottawa with 15 years of industry experience in the development and clinical launch of novel biological products, including oncolytic viruses, viral vaccines, cell therapies, antibodies, peptide therapies, and more. She has held several leadership roles in CMC, manufacturing, and QC operations, as well as in development and product scientists. Currently, she is the Managing Director of Product Development and an expert in residence with Capital Bioventures, in addition to being a biotech consultant supporting technical operations and CMC for biologic product development and clinical launches. So with that, Chantel, I'll pass it over to you. And I see you're already sharing your screen. That looks great.
Awesome. Well, thank you, Ellie. So good afternoon, everyone. I'm Chantal. And I'm really happy to be here to share some insights today on scientific and investment readiness, specifically to navigate how to navigate those early developmental stages for your therapeutic product. So the intent of this first session, or this first part, is to go over the commercialization roadmap. I'm just gonna click, there we go. And of course, this is all, a template. The nuances and details of every specific product development landscape will differ a little bit by product and by indication. But generally speaking, this is kind of how it evolves. And so here what you can see is a roadmap that outlines the typical activities, timelines, and even costs across the early product development, so all the way to early clinical proof of concept study. And what I've done is I've segmented the development into key stages. So we start with the preclinical evaluation, moving on to proof of concept, preclinical validation, through to IND or CTA submission, and then into early clinical development, kind of getting ready for pivotal trials, but I've not gone beyond that. What I've highlighted in bold is what tends to be critical path. Those things that can take the longest and trip you up. And so we're looking at things like GLP, tox studies, process development, manufacturing, and regulatory interactions. Importantly, when you look at the time and cost, it's important to note that these will vary a lot. based on the modality and based on the indication and the novelty also of the product. Generally speaking, biologics will require a lot more time and investment, cell therapies even more so. And then small molecules are on the lower end. So let's dig into the details here. We'll start with preclinical evaluation. The objective here is to evaluate your product candidates to optimize a lead. This is often through some sort of iterative evolution with a functional assessment of some sort. And within this, you're generating a preclinical data package that demonstrates the efficacy and safety in a relevant model. Next, we have preclinical validation. And sometimes even on the roadmap, this can kind of coexist or be lumped in with an IND enabling phase. Generally speaking, the objectives here are to demonstrate the feasibility of your lead candidate and the likelihood that it can become a human drug. And so we're answering questions like, is it manufacturable? Is it compatible with typical GMP manufacturing unit operations? Does it behave well in early models of toxicity? And importantly, do dose range finding or DRF studies confirm that you can dose at a reasonable level and get your expected safety and efficacy? And so all of this culminates in some sort of pre-IND or pre-CTA meeting typically. And what you're going to try to do is evaluate what your key gaps or questions are that remain in getting to your clinical product. So what does your GLP tox study design have to look like? What should your GMP manufacturing scheme be? And especially, what do your QC release specifications look like? And then also, this is a great time to propose an early clinical design. And so this meeting is an opportunity to really put these proposals in front of the health authority and get their feedback. And the reason you want this is because once you're in the IND enabling phase, this is where serious spend and time can occur. And this is also where you enter your regulated world of GLP and GMP. And these genuinely can feel like very different worlds. So now you're into serious process development. You're trying to achieve a phase 1 ready GMP compliant manufacturing process. You are into analytical development and qualification to have all of your methods for product release and characterization. You're looking at your GLP compliant toxicity studies with qualified bioanalytical assays for your analysis. And then you're actually performing your GMP compliant manufacturing of your drug substance, your DS, and your drug product, your DP. All the while refining and finalizing your clinical development plan with your principal investigators and other clinical stakeholders. And so all of this culminates in your clinical trial application, your CTA, or your IND submission. This is where you're asking the health authority whether you can move on to clinical trials. This whole phase, this IND enabling phase, extremely expensive, time consuming, and high risk, both from a technical perspective, because you're now entering a completely different technical space with GMP manufacturing and GMP compliant analytics and GLP tox studies, but also a business risk because you're going to spend likely millions and potentially years. You don't want to end up without that regulatory approval, that green light at the end. So hopefully you get the green light and you can move into early clinical development. This typically begins with a phase 1 trial. Most drugs will start with a dose escalation, starting at a very low dose, treat a few patients, look for a clear safety signal, and then being able to dose escalate from there. Some products will start with a phase 1A in healthy human volunteers. Others, just because of the indication, like cancer, for instance, you would go into phase 1B in already metastatic end stage patients. And with some products, for instance, autologous cell therapies, a dose escalation isn't really even feasible because you get the dose that you're able to manufacture. And it's patient specific. And so in that case, you're more going into like a phase 1B dose confirmation. No matter where you land on that scale, the first objective of early clinical trials is safety. That's what the regulatory authority cares about. From a business perspective, you're also looking for that first whiff of efficacy. You probably won't have the patient numbers to get statistically significant efficacy, but if you have a really solid biomarker and correlate program with some great bioanalytical assays, you can start building that case for efficacy. At this point, you may also be dealing with continued manufacturing for resupply to your clinic, and even considering process and analytical development to get ready for phase 2, especially if you need to scale up your manufacturing. And then for the purposes of this talk, I'm going to end with kind of an early clinical proof of concept study. And so once you've demonstrated safety, you've defined your dose and your regimen, typically you would move on to some sort of expansion cohort or phase 2 in a larger population to start defining your efficacy. From a CMC perspective, from a product development perspective, at this point, you're preparing for your pivotal trial. And so probably performing manufacturing and analytical tech transfers to a new contract organization that can be better prepared for your pivotal trial and early commercial. You're looking for big money raise at this point to fund a pivotal phase 3 in a large patient population. And from a regulatory perspective, you're looking at your end of phase 2 regulatory meeting and assessing what you're going to need to be ready to launch your pivotal trial. And so there is a ton on this timeline, and I'm going to show you what it can look like in an optimistic Gantt, I will say. But before I show that, I think it's important to note that the exact timeline of how this all plays out is highly contingent on these 3 resources. So number 1, do you have all your cash ready to go? Or do you have to pause to raise funds between phases? Do you have your team ready to go that not only knows the segment they're in, but is able to prepare for the next one? And so if you're, an early startup, this can actually be quite costly. And it's important to think about your team um, uh, in the totality of all the people that you have influencing you. So this can be advisors, consultants, and also your internal team. And then lastly, do you have your experienced partners and vendors already in place, contracted, ready to go? And so having all of these elements in a startup would be highly unusual. So don't think that this is the goal by any means, but it's important to understand how these resources play into your timelines when you're putting those expectations in place. And so this is a very template, hand wavy, optimistic Gantt, a timeline of how all of these things play out. But it's just so that you can understand how they stack, how they can be compressed, and where those critical milestones sit. It's extremely aspirational. It can be realistic, though, if you have those 3 resources set up in place. And so often, for a mid-sized biotech, this actually can play out this way. I also want to note the dependency on some critical vendors here. So you've got your GLP compliant contract research organization, your GMP compliant contract development and manufacturing organization, and your GMP compliant contract testing organization. And so this is where having these vendors in place ready to go is really important. And I think that's it for now for me.
That's great. Thank you so much for giving us that overview. As I mentioned, we won't do Q&A for Chantel at this time, but if you have questions, we'll have a chance to ask Chantel questions later in the agenda. So please put them in the Q&A, just noting that they're for Chantel, and then I can bring them up at the Q&A segment later. Our next speaker is Matthew Jeffers, who will be discussing how to align research evidence with regulatory expectations to accelerate cell therapy. Matthew is currently pursuing a PhD in epidemiology at the School of Epidemiology and Public Health, University of Ottawa, with Doctors Manoj Lalu and Dean Ferguson. Matthew's research thesis seeks to address optimization of preclinical experimental design and analysis to maximize validity, as well as methods of assessing preclinical evidence to better inform decisions to translate novel cell therapies to early phase clinical trials. He has 9 years of research experience in preclinical models of stroke recovery, rehabilitation, and late preclinical therapeutic assessment as a laboratory manager at the University of Ottawa. So with that, Matthew, I'll hand it over to you.
Well, thanks a lot, Ellie. So yeah, thanks for the introduction. I'm Matthew, and today I'm going to be talking to you a bit about how can we try to align early research evidence with regulatory recommendations to try to mitigate the risk of objections when we're moving cell therapies towards clinical trials, focusing mainly on that preclinical evaluation, validation kind of stage that Chantal was talking about. So I think that we're all here today with really a common interest in this idea of developing cell therapies that ultimately we'll be able to take to patients to try to help treat disease. However, a major barrier in this journey is really developing that data package that Chantal was talking about, needed to support regulatory approval to enter clinical trials. Um, we know that cell therapies experience particular difficulty entering clinical trials, and they receive double the number of regulatory objections compared to biological drugs for matched indications. And so in this talk, I'm going to be touching on a few of the considerations around these increased objections, such as challenges associated with the complexity of cell therapies as living drugs, how changes throughout development can affect the definition of a product. This really gets into Chantel's topic, I think, as well, and why demonstrating the potential benefits of a product, it's especially important for cell therapies. And in particular, I'm going to be focusing on this idea of how do we establish proof of concept in preclinical development. But just keep in mind, all these topics and challenge areas, they're all really interconnected with one another. So a lot of you in the audience, you're going to be familiar with the increased complexity of producing and characterizing cell therapies in comparison to both small molecule and biological drugs. As living drugs, cell therapies have unique potential risks as they could become persistent and permanently integrated in the patient, as well as they have really challenging logistics around manufacturing, transportation, storage. Raw materials can often be collected from individual patients themselves. And innate biological variability throughout cell therapies really makes establishing mechanisms of action, consistent manufacturing processes, identifying markers for quality assurance and potency. It's very difficult for cell therapies compared to these other drug classes. So because of these added complexities in characterizing cell therapy products, the field has generally moved in this direction of saying the process is the product. Like Chantel just told us, the manufacturing process essentially defines what the product is. So any changes to our processes give us a fundamentally different product, can lead to a need to redo studies, do additional comparison studies. And this means, particularly for cell therapies, I think as early as possible in development, we should be starting to think with the end in mind. Manufacturing products, conducting studies, even in these early preclinical phases, geared towards minimizing potential changes throughout development and really directly targeting processes and outcomes towards what's going to happen in the clinical setting. We don't want to do a lot of foundational work on what a regulator views as an apple, and then through changes to our processes, we give them an orange in the end. That can lead to these objections and a lot of time, a lot of money needed in order to be able to address them. So not only do we need to consider how to align our preclinical and clinical studies more carefully for cell therapies, We also need to consider how to establish proof of concept around potential efficacy for cell therapies more carefully than for other drug classes. Because of the increased risks and variability associated with cell therapies and their potential for permanent integration and persistence in the body, regulators place more emphasis on the need for a potential benefit of a therapy in order to justify human trials. So we see more emphasis in regulatory guidance for cell therapies on the idea of providing functional evidence for potential biological activity. And there's also a stronger need to support the therapeutic rationale of the product under intended disease conditions. However, as opposed to manufacturing, safety, clinical trials, we don't have any dedicated guidance documents for how to demonstrate this proof of concept. Regulators mainly provide guidance on a case-by-case basis. And so this lack of clarity, it can create some problems. Like I mentioned, cell therapies have more regulatory objections than other drug classes. And anecdotally, we've also had discussions with a lot of different research teams that have had to go back and do additional preclinical studies to more robustly demonstrate potential mechanism of action, proof of concept, than they'd originally planned to do when they were trying to move into phase 1 clinical trials. And so this demonstrates that researcher and regulator perceptions on what might be sufficient proof of concept, they're not always aligned. So how can researchers learn about what the regulator expectations are for preclinical proof of concept in cell therapies? It's actually fairly difficult. Firstly, although many regulators have different expectations around what evidence might be needed for cell therapies than they do for other types of drug classes, we don't necessarily have dedicated guidance documents about cell therapies. Guidance documents are generally hard to find. They can be spread across regulatory websites. We don't have a central repository like PubMed for regulatory guidance. And they can also be very difficult to understand. They can be written in carefully structured legal language. Depending on the regulator, they might only be available in a local language. And they can cover a lot of diverse topics and span hundreds of pages. Lastly, like I mentioned, there's no dedicated guidance for how to demonstrate proof of concept in preclinical studies. These recommendations, they can be intermixed with other topics. They can be spread across multiple documents, like in safety, quality assurance, clinical trials, like I mentioned before. And so it can be a bit of a needle in a haystack trying to find how should I be showing this proof of concept. So we were really interested in trying to solve this problem of how to show proof of concept. How could we make this easier for researchers? Are there any globally recognized recommendations or best practices around what cell therapy developers can do to demonstrate preclinical efficacy? Hopefully to help avoid running into this wall of regulatory objections that we face with cell therapies when trying to advance into human clinical trials. So we engaged a team of over 20 researchers around the world with combined expertise in 16 languages to try to overcome these challenges and synthesize how to demonstrate proof of concept so that individual researchers don't have to go out and try to do this themselves. So we searched for regulatory guidance documents from members of the International Council for Harmonization, which is made up of over 40 national regulatory organizations that govern many of the world's clinical trials. We also directly contacted regulatory agencies to request relevant documents, and we consulted third-party sources of international regulatory information like NIH ClinRegs to ensure that we had identified all the relevant laws and regulations for a lot of countries where cell therapy clinical trials are done. And this gave us our baseline data set that we then further searched for internal references to other guidance documents that we might not have found in our original search necessarily. In the end, we identified 1,215 regulatory documents that we then screened for relevance to preclinical proof of concept for cell therapies. So out of those 1,215 documents, we found 182 that ultimately contained guidance that was relevant to demonstrating this preclinical proof of concept. This map shows the geographic distribution of those 182 guidance documents that we included. You'll notice that the USA, European Union, and Japan, they have the greatest amount of guidance. China, South Korea, and Brazil also have large and increasing amounts of guidance in this area. Mostly that has been more recently developed. And the rest of the countries in the ICH have, including Canada, have little or no guidance on preclinical studies for cell therapies. And they seem to mainly defer to existing FDA or EMA guidance. And so this actually amplifies the problem. If you're looking for guidance around how to show preclinical proof of concept, you might not need to look at only your own country's guidance. You might actually have to go look at other countries' guidance as well, such as FDA or EMA. Your regulator might consider those countries' guidance relevant to you, and that guidance on proof of concept, it can be spread across 20, 30 documents from these regulators. So for the sake of time today, I'm going to provide an overview of the most common high-level recommendations that we saw in this 182 documents, and as well as some of the more detailed recommendations. On this and the slides that follow, I'm going to be providing a percentage of documents out of the 182 where we saw a given recommendation, as well as a percentage out of 38 regulators where we observed the recommendation at least once. So overall, we saw these 4 key areas where regulators worldwide seem to agree in how to establish preclinical proof of concept prior to translation into clinical trials. Really, these 4 items are the most globally agreed on concepts. So there's a strong need to establish a plausible mechanism of action for cell therapy from the preclinical data. We also need to be thinking with the end in mind, like I mentioned at the start of the talk, and match preclinical intervention parameters to target downstream clinical trials. We need to be thinking really carefully about our preclinical models to ensure they're clinically relevant. And we should also use outcomes in the preclinical stage that are meaningful for clinical translation. So in the next few slides, I'm going to give a bit more detail about what each of these key recommendations mean. Keep in mind, we also have a published paper about this where we give even more detail and regulator-specific guidance. So 89% of regulators suggest that preclinical evidence should demonstrate a potential mechanism of action for the cell therapy. But what do regulators mean by mechanism of action? So we identified 3 main features that commonly appeared whenever mechanism of action is discussed, regardless of the regulator. So namely, the cell therapy should be present in the clinically relevant site of the body. We should have evidence for that. The cell therapy should display functional properties proposed to be relevant to its clinical effect, such as producing relevant proteins, having relevant surface markers, etc. Also, the tissues of the treated individual should demonstrate a pharmacological response in the presence of the cell therapy that's in line with the proposed clinical effect. And this can have implications that are relevant for identifying potential potency markers or other manufacturing quality assurance markers. Ultimately, establishing these elements of a plausible mechanism of action early on are critical, because this then will tie directly into potential quality attributes for manufacturing as well. So another key recommendation across documents and regulators was that the parameters of cell therapy administration should be as similar as possible to those anticipated in downstream clinical trials. What does that mean? Well, simply, we should consider using similar dosing schedules, routes of administration, as we think are likely to be used in clinical trials. Most regulatory documents emphasize that the predominant focus of preclinical studies should be on establishing a safe and potentially effective dose for clinical trials to start at. Additionally, cell therapy products and any co-interventions used in the preclinical studies should be the same or as similar as possible to the products that are going to be used in clinical trials. So this goes back to that idea, trying to have our processes locked in as early as we can, using product manufactured with those processes, even in early preclinical studies if we can. Any deviations from that in manufacturing or delivery of products could potentially lead to evidence being viewed as irrelevant by regulators, potentially needing to repeat studies. The majority of documents also provided recommendations to ensure that preclinical models were clinically relevant to the populations and indications that they were intended to represent. So many documents suggest a package that includes both in vitro studies on human tissues as well as in vivo data using animal models. Researchers should use models that ensure feasible delivery of the target human intervention product. Again, that idea, we should be using the same product throughout development. This could mean we might have to consider using immune suppression, immune system humanization, in order to be able to use the human product. We could also have to consider using large animal species to be able to deliver therapies at actual cell doses using routes of administration or delivery devices that are going to be used in clinical trials, and perhaps, across multiple studies to achieve all these things. Overall, researchers should attempt to emulate the clinical trial population and disease as closely as possible in preclinical studies and be transparent about the similarities and differences of a given model from the human species. How do these different models go together to give us a robust sense of proof of concept? So compared to other drug classes, we see that guidance for cell therapies has a greater emphasis on the need for preclinical testing using actual models of disease rather than healthy animals or healthy tissues. So the last key recommendation that we saw in more than half the documents and regulators was that preclinical studies should use meaningful outcomes for clinical translation. So when this was discussed, the most common accompanying detail is to use surrogate endpoints or biomarkers that you can actually potentially deploy in both your preclinical studies and your clinical studies. So can we have a consistency in how we're measuring potential biological actions of these cell therapies throughout development? Other important points were that preclinical studies should establish the duration of observed beneficial effects and the persistence of cells in vivo using time points that are relevant for the proposed clinical indication and the clinical follow-up period in human trials. So we really should be seeing that these cells are sticking around as long as we think that they're going to need to in clinical trials, even as early as in our preclinical evidence. Interestingly, we observed that although 66% of regulators suggest using experimental design practices to minimize bias in preclinical studies, very few documents provide more detail as to what that actually entails. The most common recommendation that we saw in this area was to replicate preclinical findings under a variety of conditions, using a variety of models, really trying to demonstrate a consistency of effects across the preclinical evidence, not placing too much emphasis on any one preclinical study. And we found that the ISSCR guidelines for stem cell research, clinical guidance documents, and other non-regulatory guidance documents provide more in-depth recommendations for rigorous study practices like randomization, blinding, sample size calculation. So potentially researchers could supplement minimal guidance provided by regulators on how to minimize study bias with these other best practices for study design. So overall, because of the unique challenges associated with cell therapy development, it's important to really start considering regulatory guidance and aligning your development strategy with downstream trials as early as possible during preclinical development. Also, regulatory guidance, it really emphasizes the need for preclinical proof of concept studies for cell therapies, and especially in relation to establishing clinically relevant mechanisms of action, model systems, intervention characteristics, and outcome measures. So we hope that this global set of recommendations will help SCN scientists better design, analyze, and report their preclinical experiments as you move through your efforts to commercialize, translate cell therapies to the clinic, and break through that regulatory wall that we've previously seen for cell therapies. So thanks to Stem Cell Network for the LC Stream Impact Award, top-up award for my salary, helped fund this study that we did, as well as other studies we currently have underway. Thanks also for the invitation to speak about this project on the webinar today. And thanks to everyone who was involved in this project, especially all the people you can see here who helped out with finding documents and sources in the non-English languages, scouring all those regulatory websites is quite a task. So today I mainly gave a brief overview of recommendations that have really broad global agreement, I would say. But if you're interested in more details, you can find them here using this QR code for our full paper or regulator-specific guidance as well. So be happy to take any questions, and thanks a lot.
Great. Thank you so much, Matthew, for that great presentation. We have a few minutes now for Q&A. So if you have questions for Matthew, please put them in the Q&A box at the bottom of your screen. Or you can just put them in the chat if you're having trouble finding the Q&A box. Either one works. I will start off by asking one question. So I was wondering if you got a sense through your research of which international regulator is the closest to Canadian system, if people are looking for guidance from other countries, from other regulators. Do you have any advice on that?
Yeah, I think that when we're doing a project right now where we're looking at clinical trials for cell therapies that are ongoing or have been done, and what preclinical evidence was used to support moving into those clinical trials. And I think for Canada, we see that a lot of the Canadian trials that have been ongoing are often done where there's also a site in the United States, there's also a site in Europe. So I think, looking at those FDA guidances, those EMA guidances, those are going to be relevant. A lot of clinical cell therapy trials that we've done, they're often in partnership with these kind of two major regulatory organizations. So although Canada maybe doesn't have as much guidance in this area, looking at those FDA or EMA guidance can be quite relevant to us.
Perfect. Thank you. And I have one more question. While I read out the question, if you'd like to stop sharing your screen now, you can do that so that everyone
Oh,
can
sure.
see our full faces. Um, and we have one more question here, which is, given the technologies are durable, do the regulators give any guidance on timelines for preclinical studies? My observation is that they have been way too short to the point of being alarming. That wasn't my observation, that was the question askers, just to be clear.
About how short? So, yeah, I think it depends a lot on the disease and the indication of what regulators are going to view as a relevant period of time. I've talked to people that are developing cell therapies that regulators have come back and asked them to do 1 year plus studies with their cell therapies. And they did not do that before they had gone to the regulators, and now this is a big roadblock, right? So I think it's really disease specific. I think, what can you do without going to the regulators? Think about the clinical indication. What do clinical trials look like in this space? What are the typical kind of timelines they're doing? Even without going to a regulator, you can start to get a sense from doing that. But like Chantal mentioned, it's very important to engage regulators sooner rather than later. I've talked to a lot of different regulators in the course of doing this project, and they all are very happy and would like people to come to them earlier and to help solve these issues before the CTA, or the IND. It gets a lot harder, and breaks everyone's hearts when big problems come up at that point, right? So I think even if you're early in development, it's a great idea to start thinking about contacting regulator, talk about what kind of timelines do we need, what's going to be relevant here for this disease, and talking to your clinical partners as well, right? What do they think is going to be done in the clinical trial if this ever manages to get there?
Great, thank you. Um, and thanks again, Matthew, for that talk. And we'll now move to our next speaker, which is Chantal Martin coming back to discuss keeping CMC in mind during early development. So Chantal, I've already given an intro previously, so I won't repeat myself. I'll just hand it over to you to start your talk. And you're still on mute.
Yep, there we go. Okay, sorry. Okay, so I'm back and that was such a fantastic talk by Matthew. And what you're going to see is I'm going to revisit some of those same themes. Um, I don't have the amazing data of 182 guidances behind me, just experience, I guess. But it's really amazing how bang on that matches with my experience as well. So if we look again at let me just click forward, um, at this template, at this product development path. The idea here that I want to really discuss with everyone is how to plan early during this preclinical evaluation, preclinical validation time for your IND enabling and CMC goals. The reality, as we've said, is that missteps in these early preclinical phases can be extremely costly and delay clinical entry. And so what we want to do is essentially plan backwards, thinking about what that first clinical trial will look like, and making sure that our materials, our processes, our assays are all aligned with future regulatory expectations. The preclinical studies and data that are generated in that preclinical phase, it's meant to support the product and clinical development. And so any big changes in your product or your process could invalidate your early studies, exactly as Matthew said. So what I want to do is walk you through some very tangible examples here. My intent is to really dive in and give you very specific examples of where you can think about your CMC objectives within that preclinical timeframe. By no means am I covering all of the aspects, but the intent is really just to give you some of those big points. So the first one that I want to talk about is your target product profile, your TPP. This is essentially a strategic document. It is a live document. It is intended to define the clinical use of your product, the target population, the key characteristics of your therapy. And so it's bridging your scientific aims with commercial realities. So it can have an aspirational flavor to it, but it really is meant to be rooted in reality. You really want to start thinking about what your target indication will be. What do those patients look like? Do they require supportive care that will need to be tested alongside your product in preclinical development? Is there a standard of care that you should be comparing yourself to? And are these patients perhaps heavily pretreated where their immune system may be diminished, or where you're going to have some criteria for entry into the trial? Matthew discussed this route of administration. It is so important to define this as early as possible. I have seen groups have to go back and repeat critical preclinical studies because they were performed with the wrong route of administration, or even using a device that wasn't comparable to what would be used in the clinic. So an example of this, for instance, if you're going to nebulize a treatment to the lung, the nebulizer that you use in those preclinical studies, it doesn't have to be the same that's used in the clinic. Obviously, a little mouse is going to have a different nebulizer than a person, but it has to work with similar parameters, and those have to be comparable and justified. As well, you want to define your dosing regimen. So is it going to be a single dose, multi-dose? How long do you think you're going to need to dose for? So all of that is very important. And the dose itself that you're planning to achieve. Upsettingly, I have seen a company where they defined their therapeutic target dose outside of what had previously been defined as feasible for that compound in a clinical trial. And so make sure that you do your research about whether your product or a similar product has already been used in the clinic and whether there are known maximum tolerated or feasible doses that you're going to have to overcome. And lastly, based on the product and the indication, you should be able to start thinking about where this product will be administered. And this can really have huge implications. If you're looking for an at-home delivery, something that a person can take on a regular basis in their own home, then this can't be something that requires -80 or liquid nitrogen storage. It has to be stable in the fridge or at room temperature and has to be, easily administered. Versus a hospital where you can have access to a -80 storage. Okay, so the next thing I want to talk about is your critical starting materials. This is extremely common for biologics, cell therapies, regenerative medicines, that we're going to have really complex biological starting materials. This can be a virus, it could be a vector, producer cells, nucleic acids, even, a tissue that you start with. Importantly, changes in that critical starting material during development could alter your critical product attributes. And so thinking about these critical starting materials early on is quite important. And so one of these would be a vector, for instance. So if you're using a viral vector or plasmid as part of your manufacturing, any component of that vector has to have therapeutic benefit and be justified. And so an example where things can go wrong here is if your vector or virus has a reporter gene like GFP. This would not be something that would typically be allowed in a clinical setting. And I have, again, seen this example where a virus was brought through preclinical development with the LAC-Z element in it. And it wasn't until they got to their IND that the FDA stopped it and said, nope, you have to remake your virus without the LAC-Z and redo critical preclinical studies. And so that can be really quite damaging to the product development. The other one is just making sure that your vector was made in an appropriate manner. And so you want your vector or plasmid manufactured with GMP reagents and on fully tested clean cells. The way you make your vector or virus is a huge risk to contamination with adventitious agents, and the agency will be diving into the details of how this was made. And then lastly, the history of your material, how it was made, the reagents used in making it, in propagating it, the clonal selection of it should all be fully documented in reports that are easily accessible. Along the same lines, if you are using a production cell line, which typically is the case in biologics, it is common to not do your preclinical studies with a GMP master cell bank. That makes a lot of sense. But you do want your development bank to use the same source of cells as your GMP master cell bank will. Ideally, even the same passage number, same passage conditions. Things as simple as, what media or supplements you're using can have an impact here. If you require, for instance, fibronectin or collagen or feeder cells to grow your cells, they don't need to be GMP quality during your preclinical studies, but you do want to know that they are available at a GMP grade so that you won't have to make a key change in those reagents later on. And then again, here it comes down to the documentation. And this isn't something that is all that common, unfortunately, in academia or very early studies, but we really have to get in the habit of documenting our history and documenting what we're doing and with what reagents. Even if you're engineering a very common cell line like CHO cells, for instance. Regulators expect traceability back to the original parental cell line. They want to know how many passages, how was it passages, what reagents were used during those passaging. If you are building on an autologous therapy where you're starting with a patient's material, obviously this can be difficult to get that comparable starting material. There are options available, though. There are blood banks and tissue banks. Unfortunately, they're a little bit harder to come by in Canada, but we do have access to the American tissue banks. Think about how you can source that comparable starting material. If you're looking to make a cell therapy out of a tumor material or from a diseased patient, that product may look very different if your preclinical work was all done sourced from healthy tissue. And so keep that in mind, and also consider having a clinical protocol for research purposes where you can get some of that tissue to perform some of your critical preclinical work. And so Matthew said this as well. The saying is the process is the product. This is absolutely key for biologics. The way you make your product will absolutely impact those critical quality attributes of your drug. And so early on, thinking about how you're making it and how you're testing it, and making sure that it is comparable to what you're going to perform in the clinic. And the only way to know if that's comparable is to really start doing research early about what that early phase GMP manufacturing may look like. So are there unit operations that are common for your product, that you should be making sure, well-established processes that you make sure that you're incorporating. Alternatively, you'll also want to pressure test the unit operations you're using and make sure that they can even transfer to GMP manufacturing. Things like ultracentrifugation, sucrose gradient purification, performing unit operations in open containers. These are all very common in a research setting, but would be extremely difficult to bring to a GMP compliant manufacturing space. And so thinking about whether you need to change those or how they impact your critical quality attributes of your material. I think too often we forget about the analytics, especially in that early time with biologics, especially complex advanced therapies, thinking about what defines the potency of your product and tracking and trending that early can be really key. Also, what impurities are you generating in your product, and tracking that impurity profile. Often we say that in preclinical development, it's okay if your product is dirtier. What you wouldn't want is for it to be cleaner than what you're going to put in the clinic. And so the only way you can really look at that is by tracking those impurities early on and comparing it to the potency. And so, importantly, the product and the process and the analytics that you use in preclinical development do not need to be identical to what you're going to put in the clinic. That would be enormously costly. But you will need to justify that the safety and efficacy of the product you're using preclinically is comparable to the safety and efficacy that you're going to use clinically. And so thinking about that comparability early will really ease your path forward. The strength should be relatable, and the impurity profile should be very similar and have very similar components. So this is one that in a previous company of mine we spoke about often, and obviously it's a little bit tongue-in-cheek, but scientific fascinoma is this idea of always getting distracted by interesting but off-track ideas. And it's really easy to get this debilitating disease. But unfortunately, in an industry setting, any study that we're doing that isn't on that product development path can delay our clinical entry, have us go down rabbit holes that, show us data that we don't know how to interpret and have to learn more about to learn how to justify. And it costs money and human resources to make it happen. And so once you are changing your mindset to that product development path, I strongly encourage you to consider that every study should have a purpose. Consider having a study log with study numbers for every study. If a study doesn't have a purpose, I don't know that you should be doing it. I also like this idea of keeping the end in mind. So sketch out what your CTA or your IND submission will include. What studies are you going to have to discuss? What risk assessments are you going to have to show? Do you have elements within your product that are very novel and are going to take extra justification or convincing? And so we want to aim for an efficient and focused research path that supports our clinical development. Success depends on the experience and fit of both your external vendors and your internal team. And so selecting contract research organizations, contract development and manufacturing organizations that understand your product, that understand your technology and the nuances of working with it, that are phase appropriate. And so if you're going to a CDMO that caters to commercial scale and you're going into phase one, that quality system is going to be hugely debilitating. It won't have the flexibility you need in those early phases. You also ideally have a vendor that understands your health authority, that understands the regulatory expectations that you're about to walk into. And then lastly, your internal team. This is an investment. You invest in people early. It is your team that will drive you to success. You want people who can wear multiple hats and can drive to execution. And this isn't just full-time employees. It's fractional, it's contract employees, it's consultants, it's advisors, it's mentors. Surround yourself with a network of people who know more than you do and just listen very, very carefully. And if you can, if your resources allow it, consider hiring before the work is upon you, meaning 3 to 6 months ahead of time, hire the person who's going to prepare for that, who's going to make sure you have everything in place so that things happen on time and well. And then lastly, talk about quality. So I've mentioned GLP, good lab practices, and GMP, good manufacturing practices. These are compliance levels. They are legal frameworks that are required for specific work. So GLP is required for non-clinical safety studies that you present to the health authority. GMP is required for manufacturing and testing of clinical material. These are not required for most of the preclinical work that you're going to do. However, quality principles can and should guide even early non-regulated work. And so what that means is consider your documentation. Consider protocols and reports for key studies. You might not need batch records, but consider having standard methods so that you're making your product the same way each time. And it's not just, how rushed you are that day. Your reagents should be fit for purpose. They don't have to be GMP compliant reagents, but again, they should be comparable to what you're going to use when you're in GMP manufacturing. Quality oversight, so having QA is not required if it's not a GXP, GLP, GMP, GCP study. But you do want to be quality minded in your team. So maybe I'll just end with a little pitch. CBV is excited to announce that we have a new program coming in the fall called Ascent, and it's for investor readiness for breakthrough biotech. This is really for early stage therapeutic companies to be investor ready, and we are recruiting now. What you've seen here is just the tip of the iceberg. Thank you.
Excellent. Thank you so much, Chantel, for that really great talk. We do have a few minutes of Q&A for Chantel. If you have questions, please put them in the Q&A box at the bottom of your screen. And I see we already have a couple who have come through. So we have somebody who's asked, and this might preempt Greg's presentation a little bit, but I'd be interested in getting Chantel's advice too. And the question is, how is funding these very expensive translational studies feasible for academic researchers? At what stage is it necessary to think about funding sources beyond traditional research grants? And what other financial resources should be considered?
Oh, such a good question. This is such a difficult time in product development because of the resource constraints. And so early on, when you're in that preclinical evaluation stage, often that is supported by grants. There are some key grants out there that will support that kind of work. And I know the Stem Cell Network actually is fantastic for this. Having some friends and family pre-seed round typically is what's going to get you to the that preclinical validation phase. Um, Angel investors, early on can be a source uh, for getting into the IND enabling, so that really costly work. That's usually where you're going for more industry funding. So VC, family offices, dilutive funding. And so what you're going to see from Greg is, uh, first of all, you're always going to be looking for funds. In that whole timeline that I showed, you're pretty much always going to be looking for money. It will be someone's probably full-time job, just always telling the story and looking for that next source of funds, lining yourself up with people early on that can be part of that journey with you, can be really enabling. So having some fantastic backers early. Um, but yeah, usually by the time you're in IND enabling, you need to look at dilutive funding at least, if not earlier.
Okay, great. Thank you. There's another question in the Q&A that says, "Hi Chantel, thanks for a great presentation. Would you please share some advice on how to educate researchers about GLP and GMP? Do you
Oh,
have resources that you would suggest?"
it's tough. Honestly, it's a totally different world. And I come out of academia. It's uh, it is kind of crazy walking into industry out of academia, and all of these acronyms, it's like an alphabet soup. It's it is really overwhelming. There are some accelerator programs now that are trying to bridge that gap. That's one of the reasons that Capital Bioventures exists. There are others like us as well in the ecosystem that are maybe more for med device, or specific indications. I think looking into those can be really enabling, depending on the makeup of your team. Programs like SheBoot are fantastic. Obio has some great programs as well. Um, it really comes down, though, to the person wanting to dedicate the time to learning this. Um, I think ultimately there's only so much you'll learn until you just try. There's a reason that investors look at prior um, innovator experience when they look to funding. It's because it doesn't even matter how successful the last company you tried or the last product you tried to develop was. The fact that you did it once means that you've learned so much about the stumbling blocks and the consequences of your actions. And so, um, my recommendation is take every opportunity to learn, but then just try and surround yourself with people who know more than you do, and be ready to fail and try again.
Fantastic. Thank you. We just have a couple more questions here, and then we'll move to Greg. The question from the attendee is it advisable that the preclinical data be checked for its efficacy and authenticity by an external auditor before the investor embark on the investment? So I think this is a little bit about due diligence.
Oh, that's
Many
interesting.
startups fail early due to poor reproducibility.
Yeah, yeah. So I think it depends on whether you're talking from a regulatory perspective or an investment perspective. Investors will typically, especially VCs, will typically hire an external reviewer to perform the diligence, partially so that they create a little bit of wall between themselves and the information that they're looking at. But also just to have someone in the industry that knows this space well. Um, that's very common within a regulatory framework. It's not necessarily required to have an independent review of your data, um, because that's what the regulator is doing anyways. Uh, it's not something that the company has to bring to the table. I guess that's one way I'll put it. So the investor is either going to bring that independent auditor or reviewer, or the regulatory authority will perform that. But I think Matthew hit on this point of reproducibility. It is absolutely within the company's diligence to present their data in more than one model. I don't think anybody anymore will look at data out of one animal model done once and think that that's significant. And so, if you want to impress people, absolutely, you have to show up multiple models, multiple replicates, and really talk about why that is relevant to the clinical indication.
Okay, great. Thank you. And thanks again for your talk. And so we'll now move to our next and final talk, which is going to bring a lot of these concepts together from Dr. Gregory Block, who's going to discuss the alignment of preclinical data milestones and investment readiness to build a commercialization strategy. Greg is a seasoned biotechnology executive with more than 10 years of experience in the development and commercialization of novel therapeutic modalities. He's currently the CEO and co-founder of Histone Therapeutics, a biotech company at the forefront of precision epigenetic reprogramming. Prior to founding Histone, he's held roles including Senior Vice President Corporate Development at Notch Therapeutics and Director of Business Development at Astellas Pharmaceuticals, where he led business development and strategic initiatives. He holds a PhD in molecular biology from Tulane University and completed a fellowship at the University of Washington. So, Dr. Block, over to you. And I think you're still on mute.
There I am. Okay, can you hear me now?
Yep.
And you can see my screen?
Yep.
Awesome. Well, very nice to meet you all. I hope folks in the audience realize that you've really received a masterclass in regulatory guidance for cell and gene therapies. When I started my first company in 2014, we literally had no idea what we were doing. We made the first clinical grade universal donor stem cells and had never heard the term GMP before. So write everything you learned down. I'm going to reverse all the things that you heard today, because you can imagine that when you look at this kind of guidance, you think to yourself, wow, it's really got to be perfect. But in reality, it isn't. And a lot of stuff gets through the juggernaut of clinical development without the most practical considerations done. I'm going to talk a little bit about why that is today. But as I go on my rant today, I want you to focus on 4 take-home messages. The first is that raising money, especially in today's environment, is not a success metric. The success metric is success, moving something to a clinical trial, selling a company, doing something where you're actually making a difference, which can be sometimes confounded with the amount of money that is associated with how you've diluted your company. The second is around balancing aspiration versus reality. Chantel touched upon this. It's a major challenge for a CEO or a startup founder to do that. If you focus simply on reality, especially if your technology is early, it doesn't work. You need to have aspiration there. But if you're over aspirational, you're hand wavy and full of nonsense. So we'll talk about how to manage that. In connection with how we manage that as a company leader or leader of a commercial enterprise where you're spending other people's money to do something, it's important to realize that the way you communicate externally serves a dual purpose, which is how you communicate internally and how you operate and run your company. And then the last part is I'll show you a thing I call the narrative cycle, which I'm sure is in some book somewhere, but I independently invented it as a technique for managing stakeholders, both internally and externally. And I'll walk you through, if there's time, an example of what that's looked like for our startup company, Histone Therapeutics, and some of the challenges that we've faced as we've built the company. And so the first thing I want to sort of reiterate here, but maybe from a different lens, is that a biotech company is a company serving a market need. And that market need is, one, to solve problems of unmet need, but the other is to service a customer, which is large pharmaceutical companies that need to purchase technologies like ours because we are much more able to iterate and develop at a much more cost-efficient manner to bring ideas to the clinic. And so this is a typical view of what the different roles of different companies in the development of a drug look like. And you can see I've plopped histone therapeutics in the bottom left there because really, when I think about how we're building the company from a money perspective, we're a platform technology that is building things for other companies to take forward. And our goal is to de-risk not only the platform, but individual assets. This is an important thing to think about when you're building a platform company. Everything that Matt and Chantel spoke about is the path forward for a single product. If you have a platform that can execute along many products, you have to think about how you're going to handle that from a business perspective, which can be very challenging. Now, the goal of an entrepreneur in the biotech space is to realize how they can extract value from this typical process that generates returns at some point during the life cycle of a drug product. I've often described drug development as a game of hot potatoes, where if you're super early, your goal is to sell the thing to the person who will take the next stage. If you're super late, your goal is to take an asset to the clinic as fast as possible. But in both scenarios, what you're doing is you're spending money to get to a next typical milestone. So in the case of a drug screening company, or in the case of something coming out of academia that has high platform potential or is maybe not in the clinic yet, you're kind of on the bottom left there. And if you're taking a validated clinical product out of academia, or you've taken it from a canned pipeline from a pharma company, you're simply playing a different game, and you're on the star to the top right there. Now, the thing that changes is the curve along this completely made up graph. Depending on the market, things will get valued based on their perception of future value. Right now, with all the changes that are happening in the world, this is kind of a flat graph. It's not looking great. And what I mean by that is, if I'm going to spend X amount of money to get something to the clinic, should myself and my investors think that we will receive a return on value for that investment of money? And so, that's something we have to think very carefully about as we take other people's money and our own time spending towards these lofty goals. And so this brings us to storytelling. And storytelling is the way society sort of moves these things forward. And if your ultimate customer is a pharma company, where you want to sell these products to, you have to start crafting your story over the long term. And you need to start figuring out who your stakeholders are that are going to help you move the needle forward. So this little graph I took actually from a grant we wrote that wanted to, I guess, double check that we knew how internal companies work. But it's kind of interesting to look at, because there's if you have a novel platform or a novel technology and these guys are your endpoint customer, you have to think about who you're influencing. You've got decision makers within these companies. You've got R&D people who are interested in your technology but have no decision-making capabilities. You have business development teams that are on the constant hunt to formulate sort of competitive intelligence strategies. And you need to make you need to make sure the story evolves over time so that eventually these folks can be your customer. Yet at the same time, you need to make sure the story unfolds for yourself. You need to make sure the story unfolds for your own team. Otherwise, you end up misaligning expectations, and you lose people over what is a very long time to have attention. And so how do you do that? Telling your story is hard. And it really is a balancing act between what is aspirational and what is reality. It's very easy to talk about what needs to be done. And when you're in conversations with investors, everyone will have the exact same advice and talk about the sort of difficult path to your product or platform. And you have to have a view of what that looks like. But when you're telling your story, sometimes you only have 3 minutes to get that across. And you are out there looking for money, you're out there looking for employees, You have to make sure your story is clear and your vision is clear, and that when people sign up to play with you, that they're not disappointed about what they get when they find out what's happening in the real world. And so the first thing I want to highlight is that it is okay to have a grandiose vision. It's okay to want to have a nucleic acid medicine therapy that competes with small molecules. What's not okay is being ambiguous about the path that you want to get there, being so over aspirational and not actually educating people along the way and leaving breadcrumb trails that can be tracked so that you build credibility around what you're doing over time. And so people can sort of look back and say, I see where these guys have come from, what they're doing, and how they're going to make a difference to this world. The approach I've adopted both in the large pharma setting, which is, your sort of, resource rich setting, but also in the small biotech setting, which is very resource constrained, is around this thing I call the narrative cycle, which is really, in a way, very silly. The narrative cycle is basically designed to match the claims you're making against the stage of your company. And I'll contrast this in one of two ways. Let's say I have a bakery, right? And you're standing there and you're going to buy some bread. You're looking at my stand of baguettes and you're like, that baguette looks delicious. I'm going to buy it. The baker does not need to sit there and tell you a vision on like how they're going to be the greatest baker in the world. It's a simple product. It's a simple transaction. You sell things and the company is based on those sales. So someone can come in and buy that company and they'll buy it, adjusting to how they can make it better and inflation, and everyone will be happy in a transaction. On the flip side, let's say you're all aspiration and you have nothing to back up what you're saying. Then essentially you're building a house of cards, and you're trying to make these grandiose claims without communicating your progress or substantiating those claims. Now, there's a lot of companies out there. The most famous one is Theranos, whose CEO is now in prison, who had basically said, we will make this blood testing device that allows us to test with a sliver of blood, and they never actually did it. And they raised, billions of dollars of investor capital and lost it all building an entire house of cards. So how do you avoid that? You need to nail down what your big vision is. You need to have it. Otherwise, no one's going to listen to you. You're going nowhere. And you have underneath that big vision an aspirational claim. And you can go and prove that claim. You prove it with the data. We're a data-driven industry, so people listen to the data. And then once you have the data against your aspirational claim, it's no longer aspirational, and you move on. This is how you can build your work plans in addition to mapping backwards from clinical development plans, which are quite predictable. But especially if you're early, this can sometimes be quite challenging. And the thing that's kind of difficult to keep in mind, being an aspirational company with a big vision, is that the value of your company is not necessarily linked to the sales of your product. So if you're spending investor capital to bring a platform technology to the market, you might find that get priced either through a financing or through an acquisition higher than you had ever imagined it would, because someone else sees the value of what it can do in the future. And so there is, a major, that's why I call it a game of hot potatoes, because there's this sort of, buy and sell mentality that happens across the development path, and that we have to take into account as we're mapping out the risk of our businesses. So with that, and let me just make sure I have enough time. Am I good on time, Ellie?
Yep, you have another maybe 8 minutes, and then I would like to move to a couple questions before we close. Yeah.
Great. So this will take very quick. What I wanted to do is actually walk you through the story of histone therapeutics as it started and as it looks now, which I think is quite an interesting way to sort of look at the way investors have pushed us in the right direction, and where we sort of fell short as scientific founders at the beginning. And so you can see here we have this title slide with a catchy phrase, reinterpreting the genome, which is completely generic. And then a statement that says our patented technology targets any gene for de-repression, creating custom gene expression profiles, yada, yada, yada. Point is, it's a lot of words that accurately describes our technology. Now, we were really impressed by our own technology. It was an AI-designed protein that inhibited another molecule called EED. So we walked through the very detailed mechanism of action of how its target molecule works in stem cells, and how it was designed in a computer, and how when fused to dead Cas9, it can turn specific genes on. Awesome stuff. And then we went even deeper into little light switches and diagrams of the epigenome, off being repressed, on being active. And then, if we look inside the human body, we're going to put a Band-Aid on it. Super clear, right? No, not super clear. It's very hard to capture people's attention with nitty-gritty details on the scientific vision. Folks don't quite get it. They're not knees deep. They need to understand what the big vision is there and how you're going to get there. And so if you contrast that with the way we tell the story now, we call it a precision therapy for genes, which I'll get into in a second. But we lay out the very big view of why do we even care about the epigenome? Why do we even care about what we're doing? And how do we make it about someone's own self? And really, we came up with this concept that's not really unique to histone, but it captures your attention, which is that your genetic code does not determine your fate. And given that we're all going to eventually die someday of our epigenetic changes, it therefore is worth it to think about how we're going to intervene with it. But all the approaches that allow us to intervene these days with the epigenome basically are very tough to digest from the perspective of from the regulatory perspective, really, that you've just heard. And so the question becomes, can we do this in a way that is precise, yet simple and druggable? And so we came up with a blueprint to do this, both with our original in-licensed proteins, as well as the one we've designed in the company. But essentially, we designed them to meet certain specs that allowed us to do something that was more drug-like and less gene therapy-like. And that led us to our catchphrase, a therapy for genes, not gene therapy. So that is the big aspirational goal right there. And so we have to start mapping that to the deeper understanding of the market. If we're doing a therapy for genes, not gene therapy, well, aren't other folks out there doing epigenome editing? Why are you unique? And actually, if you take the principles that Chantel and Matt talked about, and you look at all the other epigenome editing companies that are out there, and you say, well, let's say you consider gene therapy to be anything that does something permanent to you, as opposed to thinking about it just as a DNA base pair modification, then in fact, the entire competitive landscape looks different. If you have a red box here on this simple diagram, you're a gene therapy, and look, we are not. And so I've made a pretty darn, a pretty bold claim, and I'm going to have to back that claim up. And the way we're doing this is we're essentially putting epigenome editors into lipid nanoparticles with an mRNA and making sure that they have no off-target effects and that they're very simple and they can be treated in a reproducible manner like a drug. And we also highlight the fact that, the way we're going to do this is not by silencing genes, but, we're going to turn genes on. And so this is helping. So we have our bold aspirational goal that we've made, and we're defining a unique category for the company. So we're trying to grasp that attention for everyone involved in the company and externally, and that this is what we're doing. And then we have to say, well, what is the claim that we're making? We've made the claim that we can do gene therapy. The claim is we can do genetic upregulation without a virus, with using a simple mRNA and lipid nanoparticles, and that turning genes up is a thing you can do, and there's benefits to that. So now I need data to back up that claim. And we now have data to back up that claim, and it's quite fascinating. We show in animal models of disease that we can turn on therapeutic genes. There it is right there. And more fascinating, the way we deliver it, these things go straight to the liver. And we can see that they drain to the liver by looking at where our construct is. But in fact, we're able to dial for the first time without a DNA promoter, we're able to dial tissue-specific gene expression, which was one of the claims we made on our front slide. And so we're able to show that this is like, wow, we can actually do the things we say we can do. But this is a non-confidential sort of teaser. It's designed to give you the sort of, if we're presenting this externally, it's like, hey, you should probably talk to Histone Therapeutics and hit the green button on your end. And so we also, we have this slide that highlights the momentum along the typical clinical path. We've done all these things from a business perspective. There's momentum happening here. And then we introduce our team, which is a critical part. And our team includes folks that are, both new to the industry and folks that are absolutely legendary. The CEO of Blue Rock is on our board. The guy who brought Atiplursen to the market is on our scientific advisory board. The guy who made the first cell therapy in the clinic is on our scientific advisory board. David Baker, who just won the Nobel Prize, his wife, who co-invented the initial AI licensed protein with us, is on our scientific advisory board. And this helps sort of cement that credibility story you're telling and hammer it home. And then we just say thank you and we're done. And with that, I'm going to say thank you and I'm done. And let's go to questions.
Excellent. Thank you for that great presentation. And we do have a few minutes of Q&A now. So if you have any questions for Greg, please put them in the Q&A box at the bottom of your screen. If we go a little over the half hour, that's okay. I see one question here, and it's actually from Chantal. Did you want to ask Greg the question directly?
Sure, be happy to. That was so fantastic. How do you evolve or change the story based on the stakeholder? I've heard this often that speaking to an angel investor, they aren't the same as speaking to, a VC, for instance. Do you find that how do you pivot that story?
Yeah, this is a really interesting question. And it's the way you do it is by having everything you talked about mapped out and in your mind. No matter who the stakeholder is, I find they benefit from the high level vision. If you start with the smartest person on the planet and they don't have the high level vision, they're going to skewer you. If you're talking to an angel who has no idea what they're investing in, what they like your hair, they still need to they still need to have it. I had a really intense experience this week in Boston last week where I met probably one of the smartest people I'd ever interacted with. And he was ripping me a new one. But I started with the high level vision and then I backed it up. I backed up all my claims with the data that I knew we had and, the difficulties that I knew we would face and ended up winning them over. So it is about starting at the beginning and just knowing when to layer on the next piece of data. But if you don't have your TPP built, you're going nowhere. You can't tell the story in a credible manner. So it's, both you have to be kind of like silly, dumb storyteller, but at the same time having the sort of preparation from your team to back it up.
That makes a ton of sense. Thank you. It's great advice.
Great, thank you. I have at least one more question, and we might have time for one more after that if anybody wants to pop anything else in the Q&A. So several years ago, maybe 4 to 5 years ago, we saw early stage biotech companies that would close very large financing rounds compared to now where it looks a bit different. Do you have any perspective on what's changed over the last few years? Where have those companies ended up, and are there any lessons learned there?
Yeah, that's a big one. So in the pandemic realm, a lot of money started going into biotechs. Investors couldn't even figure. They're like, I'll just throw my money in it. They weren't even. It's like a hot housing market where people are buying without inspections. And so a lot of money went into companies, and the investors actually exited those companies with incredible returns. And that's when I talk about the game of hot potatoes, that's what I'm talking about. You can flip a technology like you can flip a house if the market is right. And if your partner is right, they'll continue it. And if the partner is wrong, they won't be able to continue it. During those, sort of frothy times, we saw companies that started with enormous amounts of capital going public very quickly, just based on stories or in licensing B-list technologies. and they did quite well. Nowadays, you have to be rock solid. I've outlived some of my competitors with literally thousands-fold less money. And so when I talk about money raised not being a success metric, I boast about how little money I've spent to actually move my data forward and build a healthy company with employees that are loyal and with a technology that is amazing. And I've not diluted myself to high heavens, and I've not taken on, built a 20-person finance team for a company that needs to reach a preclinical milestone. And so I think there's a right sizing happening in the industry right now, but it is putting a lot of downward pressure, which I hope will generate some gems, and I hope we'll be one of those gems.
Awesome, thank you. I see one last question in the Q&A here, and it's actually to all panelists, which is perfect. And it's, what one piece of advice could you give an academic researcher that's embarking on a translational project towards a clinical trial?
I can start. My advice is surround yourself with a network of people who know more than you do. Don't think you know this world. It is very different, but don't be scared to jump in.
Awesome, thank you. Matthew or Greg, did you have anything?
Yeah, for me, I think I worked in academia for a long time, in this kind of like translational space. I think it's so important to really engage with the clinical environment, like have people that you collaborate with that are clinicians, so that you can actually understand what is the need in the space. How does this technology or product I'm building fit into continuity of care? I think that's so important, not only in terms of getting a model that makes sense and actually maybe has some validity to what's going on in the clinic, but also just having a product that actually can fit into the environment somewhere. So I think really, really think about the clinic and, if you're not a clinician yourself, partnering with those kind of people.
Yeah, and
Awesome,
maybe I'll add that I think it's really important to think about your business model. I think when you're taking outside money to build a product, you have to be able to sell that product one day. And a lot of these really complex therapeutics are not products. They're not, feasible from a market perspective, but they need to have a place. And so I think we need to think about this not only from the industry perspective of how we like deploy, investment money, but also how we employ, especially in Canada, our own public money. I want to see our public money going to helping Canadians. I want to see rare disease populations in Canada being able to be treated without having their drugs withdrawn. There's some things that I don't think are money-making ventures, and I think are services to society. I think a good corollary to that is, I have not seen a company that sells organs if you need an organ transplant. And some of these complex therapies are more akin to that than an actual sellable product.
Great, thank you. So as we close, I just wanted to take a second to give everyone a sneak peek at what's coming up next for the Stem Cell Network. I hope everybody can see this slide. In the fall, we'll be launching our Incubation Investment Awards, so hopefully that'll be of interest to lots of people who are on this webinar today. The QR code is there to be signed up to to get a notification of the August informational session. And this will be a new initiative to further Stem Cell Network's translational mandate and position regenerative medicine innovations for commercialization readiness. This will be an early stage investment vehicle. The investments will be up to $1 million over 3 years. And additional expertise will also be available to assist startups. So the application process will open in fall 2025, and the awards are planned for spring of 2026. So I hope everybody keeps an eye out for that. And then with that, I'll officially close. I want to say one more big thank you to all of our speakers today, to Greg, to Matthew, to Chantal for taking the time to educate us about embarking on this commercialization journey. Thank you for everyone for attending today. Please take a minute to fill out the post-event survey. The QR code for that is there on this slide, and you'll also get a link in the follow-up email. We really want to hear what you thought about this event, and then also what kind of events you want to see in the future. And then always on our website, you can go to find out about other training opportunities that we're running. So thank you very much to everyone once more time, and have a good afternoon.